Okay, so today we'll talk about the cellular level. You know that one step or level higher or bigger or larger than the chemical level is the cellular level. So, in today's lecture, first I'll talk about the definition of a cell, what a cell is. Then we'll see the parts of a typical cell. What are the parts a typical cell should have? Then we'll talk about the structure of a typical cell. First, we'll talk about the structure of the cell membrane that gives the boundary of the cell. We'll see the structure and function of the cell membrane. Then we'll talk about the proteins in the cell membrane. If you see the structure of a cell membrane, you'll see cell membrane is a phospholipid bilayer, but in which protein structures are embedded. So, what are those protein structures and what functions they perform there? We'll see that. Okay. Then we'll talk about intercellular junctions or joinings. Intercellular junctions or joinings do what? Hold the adjacent cells together. So hold the cells together by forming attachments. So we'll see what are different types of cell junctions present in our tissues. Then we'll talk about the transport across the cell membrane. You know that cell membrane is a semi-permeable membrane. That means it is partially permeable. It will allow some chemicals to pass through it, but not everything. Selectively permeable. Make sense? So it allows some chemicals to pass through it, but it will also block some chemicals. So we'll see what are the processes of transport of chemicals across the cell membrane. Okay? Different ways the chemicals can pass through the cell membrane. We'll see what are those different ways or processes. Then we'll see the response of a living cell in three different types of solution: hypotonic, isotonic, and hypertonic. So if you take a living cell, for example your red blood cell, and put in three different types of solution, how the cell will respond in those three environments. We'll see that. So those are the things we'll cover in this lecture. First, what is a cell? The cell is the smallest structural and functional unit of living organism or living body. Okay? The human body has 50 to 100 trillion cells. Just think that it's not million or billion, trillion cells. Over 20 different types of cells are found in human body. They are different. How they are different? They are different in shape or size or their functions and their structures. So they are different from each other in many ways, by size, by shape, by structure and function. But all cells chiefly composed of CHO and N. You remember in last class I mentioned that over 96% body mass is formed by these four elements, right? So these four elements are the chief components of the living cells, CHO and N. Now, why we say that cell is the smallest structural and functional unit? You can keep a single cell alive. Scientists are doing all the time. Take your brain cell or any cell and put in a petri dish in solution, give oxygen, nutrition, the cell can stay alive. You can keep the cell alive. You know that. Another example, bacteria, virus, amoeba, those are single cell microorganisms, right? And they stay alive, they do their function. So, cell is the smallest structure that can perform its function. But if you break the cell membrane, it will die. It will not perform the function. Make sense? So that's the smallest structural and functional unit that can individually perform its function.
I was just going to say, what about the mitochondria? It's got the double bilayer.
Mitochondria has cell membranous organelle, but mitochondria alone cannot do the task. It needs help from other organelles and cytoplasm around. So it will not stay alive once you break the cell membrane. When the cell membrane is cell is lysis, that means the death of everything. No organelles will stay alive independently, but you can take a cell and keep it alive. Okay? Because inside the cell you have all the materials to keep it alive. They work together. It's not like, but once you take mitochondria out, it cannot work alone. Make sense?
I was just thinking of the theory that they thought it used to survive by itself and then it was engulfed.
Engulfed what?
That's what. I was just thinking that, I've heard that they think that it was engulfed way back in the day. That's why it has a double bilayer.
Oh, so yeah, talking about evolution. Those theories are, scientists are divided about those theories. Like initially, evolution was very, popular, and everybody accepted that. Then, like Darwin theory, those kind of theories are now scientists are getting divided. That a lot of things are still not proved how the things happen. Like you are saying that mitochondria was engulfed and it was taken inside the cell and become a part of the cell, right? Something like that. Those are theories. But proof is that if you break the cell membrane, it will die. Okay. So, how the cells are different? We know that over 200 different types of cells are present in the human body. Here is some examples how they could be different. You see the top left, two fibroblasts, connective tissue cells. They produce fibers and look different than the red blood cells in the right side. Red blood cells are round, flat, and color is red, so appearance is different, and also function. Red blood cells transport oxygen. So you see they are different in shape, size, appearance as well as function. Red blood cells don't have nucleus, so structurally it is different. Next one you see epithelial cells. They are also different. Then go down, you see two different types of muscle cells. Even though both are muscle cells, but they are different. First one is a skeletal muscle cell. It is very long and many nuclei inside it. You see that, many nuclei. And in the right side, you see smooth muscle cells, short, small, and single nucleus. Then, if you go down in the bottom left, you see the fat cell or adipose tissue cell. The nucleus is located not in the center but attached to the membrane. And right side, macrophage. It's a phagocytic cell. Macro means big, large phagocytic cell, and it has many processes, extensions from the cell membrane, and they love to engulf the antigens, microorganisms or dead cells. Then right top, you see a nerve cell or a neuron. Neuron has a cell body, and you see from cell body a long process and a number of short processes come out. The long process is the axon, short processes are dendrites. So neuron looks completely different than other cells, and function of a neuron is electrical signal transmission. And the last one you see is a sperm, male sex cell. And a sperm looks like a baby fish, right? Has a head, body or midpiece, and a tail. And by moving the tail or whipping the tail, it can swim. So, and function is fertilizing the egg. So you see a few examples how the cells are different from each other. Okay, now we'll see the parts of a typical cell. A typical cell has a cell membrane, which is also called plasma membrane, that gives the boundary of the cell, forms the boundary. Then you have cytoplasm, which is the fluid that cytoplasm consists of fluid and cellular organelles. So fluid and cellular organelles are present in the cytoplasm. The fluid is called cytosol. So cytosol is the fluid. So I can say cytoplasm consists of fluid, which is called cytosol, CY, and organelles. Okay, organelles. Now, organelles, there are two types of organelles in cytoplasm: membranous and non-membranous organelles. Okay, so some organelles have membranes, outer covering. Some organelles don't have. Then the nucleus, usually located in the center of the cell, and nucleus is the control center of the cell. Why? Because the genetic materials, DNA, are located inside the nucleus. Okay, so DNA are located inside the nucleus, and DNA controls the functions of the cells. So that's the control center of the cell. So those are three parts of a typical cell. Why I said typical cell? Because you know that some cells don't have nucleus. For example, red blood cells. Red blood cells don't have nucleus. So that's not a typical cell. Typical cell means those cells have all these three parts. This is a picture of a typical cell. So you should be able to see those three parts. First, let's see the outer boundary, that is the cell membrane. And the function of the cell membrane is, number one, Cell membrane gives the boundary, I have already mentioned. It gives the shape of the cell. Cell membrane separates intracellular fluid from extracellular fluid. So separates intracellular and extracellular fluids. That means inside and outside the fluid are separated by the cell membrane. Uh, Now, if you another important function of cell membrane, you can write it down, is it helps to recognize the nearby cells. So the cell membrane helps to recognize the adjacent cells, nearby cells. So that is another very important function of the cell membrane. Now, how many functions I have mentioned? Two, three, or four. I think four. Number one is boundary. Number two is giving shape. Number three is separating the intercellular extracellular fluid. And number four is what? Recognizing the nearby cells. Next cells. Okay. Now, if you see the cytoplasm, which is located between the cell membrane and nucleus, in between the cell membrane and the nucleus. And you see that cytoplasm has a number of organelles. I'll just mention a few organelles and their functions. You're supposed to learn these in intro bio, but just quickly going over. So, you see around the nucleus, the blue colored tube-like structures, those are endoplasmic reticulum or ER. Now, they are located around the nucleus. Now you see these endoplasmic reticulums have ribosomes attached on the surface. These tiny dots, these are ribosomes. So ribosomes are attached to the surface. So these are called rough endoplasmic reticulum or rough ER because the surface is not smooth. Rough endoplasmic reticulum because the surface has ribosomes attached on it. And another part you see here on the endoplasmic reticulum, no ribosomes are attached. So the surface is smooth. So these are called smooth endoplasmic reticulum. Okay, so smooth here. Now, I'll just mention a couple of important functions of rough and smooth ER. First, rough endoplasmic reticulum. Modification of proteins. Protein modification. So, after the protein is produced, then or synthesized, then modification is done by rough ER. Not only that, folding of proteins, another function of rough ER. Then transportation of protein. So it deals with protein. Those are three important functions of rough ER. It modifies, folds, and transports proteins. Okay. Then smooth ER performs other important functions. Some other important functions are performed by smooth ER. Number one, detoxification. very important function. Detoxification. Uh, the harmful chemicals that you take from outside or produced inside the body, many of those harmful chemicals are detoxified, neutralized by inside the smooth endoplasmic reticulum. For example, we take medicines, right? And you know that all medicines have good as well as bad components in it. So toxic elements. So those are neutralized inside the smooth ear. Number two, uh, synthesis of lipids. Lipid synthesis is another important function. Number three, storage of calcium. Okay, so those are some important functions performed by smooth ear. This part. Okay, now the functions of ribosome. Ribosomes are those you see tiny structures. Many of them are attached on the surface of rough ear. So ribosomes, just know that ribosomes are produced in the nucleus and then sent to the outside. And ribosomes are responsible for manufacturing protein. So protein manufacturing or synthesis is the function of ribosome. Okay. So manufacturing of proteins. Then we'll see these green structures. You see here, both ends are tube-like structures piled together, but both ends are expanded or round, kind of round. So these are Golgi apparatus. Golgi apparatus does what? Packaging of proteins. Number one. Number two, formation of vesicles. Okay, vesicles. By forming the vesicles, the Golgi apparatus sends the chemicals to the outside, protein chemicals to the outside of the cell. You see here, what happens, the protein protein molecules go to the both ends of those tube-like structures in Golgi apparatus, and then the ends get detached from the main body and form the vesicles. And then vesicles go to the cell membrane and rupture. That is called exocytosis. And the chemicals or protein molecules are released to the outside of the cell. So we can say that by producing vesicles, Golgi apparatus helps to send the chemicals to the outside of the cell. So those are the important functions of Golgi apparatus. So now if I start again from the beginning, ribosomes do what? Make proteins, manufacture proteins, right? And then give the proteins to the rough endoplasmic reticulum because ribosomes are just sitting on it. So we'll send the protein inside the rough ear. And rough ear will do what? Modify it, fold it, right? And give it to the Golgi apparatus. And Golgi apparatus will do what? Do the packaging. And will send to both ends and produce vesicles. Make sense? And then vesicles do what? Take the protein chemicals to the outside of the cell membrane, released by exocytosis. It's like a factory. In a factory, what they do? They produce, right? Then they do the modification, folding, and then packaging, then sent. Make sense? So your cell is doing similar functions. Now you see the orange color structures, those are mitochondria, very important organelles. Mitochondria is the primary site for cellular metabolism. Primary site for cellular metabolism. That means metabolism mostly occurs inside the mitochondria. And metabolism, that produces ATP or energy. So that's why most of the ATPs are produced inside the mitochondria, and mitochondria is called the powerhouse. Makes sense, right? Powerhouse of the cell, because most of the ATPs are produced inside the mitochondria. Nucleus. Nucleus is the control center. It has two parts. Outer covering is called the nuclear envelope, and inside part is called nucleoplasm. So nuclear envelope and nucleoplasm. Nucleoplasm and nuclear envelope. Okay, so those are two parts. In nucleoplasm, you have the nucleolus. Nucleolus is small round structure inside the nucleus. Okay, and inside the nucleolus you have the DNA. Okay, so the genetic materials are stored, highly condensed there inside the nucleolus in chromatids. Okay, so just know those few important things. Then we'll move to the plasma membrane or cell membrane. Plasma membrane is a phospholipid bilayer. If I ask you, tell me in one sentence, what's the structure of the plasma membrane or cell membrane? It is a phospholipid bilayer in which proteins are embedded or inserted in it. Now, I have already mentioned the functions of the cell membrane or plasma membrane. There is a theory, or we say model, to define the structure of the cell membrane or plasma membrane. That theory or model is called fluid mosaic model. Why we say that? If you see a cell membrane under electron microscope, very powerful, 1,000 times if you make it bigger, okay, and see under the electron microscope, what you will see something beautiful. Beautiful means it's like a beautiful floor, white floor in which you have marbles. Marbles, beautiful rocks or marbles are inserted in that floor. Not only that, the floor is moving like this. Why that happens? Phospholipid bilayer is the white lipid layer, and the proteins are inserted. Proteins are like beautiful marbles are inserted in it, and what happens? Those proteins are always changing their shape. Okay, changing the shape. So if you see that under the microscope, you'll see the proteins are moving, right, in white bed. So that's why it is called the fluid mosaic theory. Okay, so the proteins, there are two types of proteins in the plasma membrane or cell membrane: integral proteins and peripheral proteins. And both proteins perform some important functions. Okay, so integral proteins are those proteins pass through. If this is your phospholipid, let me draw this way. This is a phospholipid layer. This is another phospholipid layer. So phospholipid bilayer. And in phospholipid bilayer, the proteins are embedded. So some proteins are deeply inserted like this. This is the protein, okay? And then you have the phospholipid bilayer, okay? And you'll see in some places the proteins are passing all through the cell membrane. Those are called integral proteins. So if the protein structures or proteins are deeply inserted or passing all through the cell membrane, those are integral proteins. Integral proteins. Okay. Now, sometimes you'll see the proteins are loosely attached to the integral proteins. These are not inserted in phospholipid. These are peripheral proteins. Peripheral proteins. Okay. So those are two types of proteins present in the cell membrane. Integral proteins help in transport of chemicals across the cell membrane by forming channels or acting as carrier. Many integral proteins work as receptors. They work as receptors. Uh, and many integral proteins work as enzymes. So help in chemical reaction. So I will show you how the integral proteins help in transporting the chemicals. In some integral protein, you will see through channels, not in all. In some integral protein, you have channel like this. So the molecules smaller than the size of the channel will pass through it. Okay, will get in or out because they have two channels. So by forming channel, integral protein helps in transport. Some integral proteins do what? Say this is the integral protein. So it will first let the molecule get in and will get attached to the integral protein like this, and the integral protein will change the shape and will move like this and drop it inside the cell. Make sense? So first it will let the molecule sit on it, on the receptor binding site, and then it will change the shape to take it inside. That is how they work as carriers. Um, some integral proteins work as receptors. What is that? Uh, this is the integral protein, and some integral proteins have binding sites for the signal molecules or chemicals. For example, this integral protein has a binding site like this. This one has also a binding site like this. This one has a binding site like this. So these are three different integral proteins. All of them have binding sites for what? Chemical molecules that will work on the cell membrane on the cell. So what are the chemical molecules? You know, you take medicine, right? Medicines are chemical molecules go into your body, and medicines must work on the cells, right, of your body. Medicines must work on the cells of the body. Is it clear? Or hormone. Hormone works on the cells, right? So hormones are medicine molecules, for example, and if the hormone or medicine molecule is, that is called the signal going to the cell membrane. Signal molecule. Okay. So Now the signal molecule will work on the cell. So first it has to bind with the integral protein of the cell membrane. So this one will come here and we'll try to find a good binding site. So which one is good binding site for this? This one, right? Because the shape and size fits. So this will come here and will perfectly bind here. Make sense? If it comes here, no perfect binding. Comes here, no perfect binding. Make sense? So when that binding signal receptor, this integral protein is receptor, binding will be perfect, then what will happen? The signal will go to inside the cell. This is inside the cell, cytoplasm. Signal will go there. Make sense? So it is like, you put the key in your car and then it starts, right? So if you put wrong key, what do you start? You won't be able to start the car, right? So the binding must be perfect, like key lock type binding. And when that will happen, the signal will get in and intercellular metabolism will start to work. Okay, so intercellular machineries will start to work. So you see that very important function by acting as receptors that will receive the signal molecules. Okay, so that is another important function of the integral proteins. Peripheral proteins, just know that I'll mention two important functions of peripheral proteins. Number one is cell to cell recognition. What is that? The cell can recognize the next cell, which cell is next to it. That is cell to cell recognition. Cell to cell recognition. You all know that your cells can recognize other cells. Do you know that? Like if you get burned in your skin, if I take skin from another part of your body and implant here, your body will accept it because these cells know that the other cells are from your body. Make sense? That is cell to cell recognition. But if I take another person's skin, most of the cases the chance is rejection, right? That is also tells that your cells can recognize the cells, right? Other cells. And that's the function of your peripheral protein, one important function. Another important function is forming intercellular attachment or junctions or joinings. So one cell gets attached to the next by the peripheral proteins. So peripheral proteins form intercellular junctions. So just remember those two functions of peripheral protein. Those are very important functions of the proteins. Here you see the structure of the cell membrane. You can see the phospholipid bilayer. This is one phospholipid layer, this is another phospholipid layer, and these are the proteins. So these are integral proteins, this one, this one, this one, and these are peripheral proteins. Make sense? Okay. And also just know that you have glucose and cholesterol in small amount. So these yellow structures are cholesterol and these are glucose. Now you see here, this green structure is what? Glucose, very good. And this one is protein, right? So together form what? Glycoprotein. Makes sense, right? Glyco is glucose and protein. Now this yellow one is what? Cholesterol, right? Lipid. Cholesterol is lipid or fat. And this is green is glucose. So together form glycolipid. Make sense? So when you join them, they join together, they form glycoprotein, glycolipid. Okay, so that is the structure of the cell membrane. Mainly phospholipid bilayer and proteins. How many types of proteins? Two. What are those?
Integral.
Peripheral. Okay, integral and peripheral. And a small amount of cholesterol or lipids and glucose. Make sense? Now, if you see one phospholipid unit or molecule, it is like this, right? It has a head and a tail. So this is a phospholipid molecule. It has a head and a tail part. The head part is also called polar end, and tail part is called non-polar end. Okay. So that is the structure of a single phospholipid. You see, if you just take one phospholipid molecule or unit, it has a head and a tail part. The head part loves water or likes water. That's why this part is called hydrophilic. Hydro means what? Very good. Hydrophilia means what? Philia. Liking. Attraction. And the tail part doesn't like water. So this part is called hydrophobic. Phobia means what? Fear. Scared. So hydrophobic. Now you tell me, do you think water would be able to pass through this phospholipid bilayer?
Yes.
Why? What do you think? Water will be able to pass through it or not?
Because the whole head is around top.
Yeah, but this whole part is what? Hydrophobic, right? Dislike water. So the water will not be able to pass through the phospholipid bilayer because the middle big part is hydrophobic. Make sense? Water will not be able to pass through. Then how the water will get into the cell? Do you remember I told you that channels are present through the integral protein, right? The channels protein will let the water molecules get through it. Okay, so proteins have no problem. Water can get through the proteins, but cannot pass through the phospholipid bilayer. So you see how the integral proteins transport the chemicals. I told you that water and water soluble molecules will pass through the protein structures because water and water soluble things cannot pass through the phospholipid bilayer. So you see two different integral proteins are transporting the chemicals in two different ways. One is by forming the channels. That channel is always open and the size of the molecule is smaller than the channel will pass through it. The first one, the channel will always stay open, so it will let the molecules pass through it. Another one has gated channel and a binding site. Where is the binding site? You see in the middle here, this is the binding site. So what will happen, I already showed you, the molecule will get in and only the molecule will be able to get in when the door will be opened. So first the gated door or channel should be open. When this door will be opened, then the molecule will be able to get in and the molecule will bind here, sit here. It's like, you have a seat, you are going there and sitting on that, then the protein will change the shape and will release it. Make sense? So first the gate will open, the molecule will get in and sit there, then the gate will get closed and this side will get open. Make sense? So it will drop it. So that's how some integral proteins transport. So first one doesn't need ATP or energy, but second one needs energy to open and close the door. Receptors, I have already explained here how integral proteins work as receptors. Some integral protein works as enzymes, help in chemical reaction. I have already explained to you the function of peripheral proteins, cell-to-cell recognition, and intercellular junctions or joints help to attach the cells together. Now, three types of intercellular junctions are most common in our body. What are those three types of junctions? Tight junctions, desmosomes, and gap junctions. So those are three common type of intercellular junctions formed by the peripheral proteins. Tight junctions prevent fluid and most molecules from moving between the cells. So the seal is very strong. So most of the molecules and fluid will not be able to pass through it. Strongly sealed, heavily sealed. Desmosomes are the junctions that hold the cells very strongly. How? You know, in your clothes, sometimes you use zipper in your pant or shirt. Uh, so desmosomes work that way. So linker glycoproteins come from both cell membranes and get attached like this, very strongly anchor the cells together. Make sense? Very strongly put the cells together. Gap junctions. Its name is telling you gap junctions have many channels or pores through them. So the molecules or fluid can go from one cell to the next, can pass through the gap junction. Okay, so it allows the small molecules to pass through. So those are three most common type of intercellular junctions. The picture here is showing the a tight junction. Uh, if you see how a tight junction is formed, you see the lower picture here, uh, two cell membranes. This is one cell membrane, this is another cell membrane. They are attached to each other, and tight junctions do what? Stitch them. Like you put two pieces of clothes together, two sheets, and stitch. Okay, like that. You do that, right? So that's how the tight junction seals. Desmosome, inner side of the cell membrane has a plaque. So you see, this is the cell membrane, and inner side of the cell membrane you have a plaque from this side, and in this cell membrane also you have a plaque in the inner side. Plaque are like disc-shaped protein structures strongly attached to the cell membrane. Then from the plaques, linker glycoproteins. These are very important. Linker glycoproteins, also called cadherins. Cadherins, it is there, or linker glycoproteins. They arise from both plaques. So coming from both cell membranes. And they, as I showed you, get attached to each other like this, very strongly hold them together. Make sense? So you won't be able to separate. Where you will find plenty of desmosomes, you will find plenty of desmosomes in the skin. And you know that sometime when your skin gets too dry, you can peel the skin. You know that? A chunk of skin will come off. Why? Because the cells are strongly attached to each other by desmosomes. So in some areas we have more desmosomes. That means the cells are more strongly held together. Make sense? And in your heart muscle, you have lot of desmosomes. Why in the heart tissue you should have a lot of desmosomes?
You don't want it to fall apart.
Yeah, you know that when heart contracts, all muscle fibers or cells must work, right? And contract together. So if they are held together strongly, they must work together, right? If they are separated, then the work will not be together. It's like, if you are standing here, 5 or 6 of you, and so like some, storm comes, like air forcefully comes, you will get separated. But if you hold your hands together, you will not get separated, right? So in the heart muscle, the fibers or cells are strongly held by desmosomes. That's another location. Gap junctions. You see, in gap junctions, there are through channels. So from this cell, the fluid or molecules can go to this one, and from this side can go to that one. So they allow the molecules, they allow the cells to talk to each other. So the cells can quickly talk to each other. It's like, if the rooms, you have many rooms and they are completely separated from each other by wall, then you have no contact to the next room, right? But if you keep many doors open, so you can easily communicate. Exactly like that, the cells can talk to each other or communicate to each other uh, through the gap junction. Just write it down. Many desmosomes and gap junctions are present in the cardiac muscle. So the muscle fibers stay together and can talk to each other all the time, which is very important to perform its function. The cardiac muscle has a lot of what?
Desmosomes and gap
Can gap junctions. Okay, that makes sense, right? Because heart muscle has to work at like a single unit. Transport across the cell membrane. Since cell membrane is a semi-permeable membrane, it allows many chemicals pass through it. So the transport occurs in two processes, passive process and active process. So passive and active transport. First, I'll tell you the main differences between active and passive. Active processes require energy, ATP, utilization of energy or use of energy. Which one? Active, right? Active one needs ATP. Passive doesn't need ATP. Active, since it requires ATP, occurs only in living cells, living cell membrane, because ATPs are produced only in the living body, living organism, right? ATP is not produced in non-living body. So two things, requires ATP and occurs only in the living body. Passive doesn't require ATP and it can occur both in living and non-living membrane. So those are the main differences. Yes.
What do you mean by non-living? Can you give an example?
If in the lab you put a filter paper and separate two solutions, then the fluid will move, right? The molecules will move. But in that case, ATPs are not being used because there is no ATP there. And it happens in nature all the time. You know, there are different density of things, right? Even in the ground, different densities and the molecules are moving all the time. But in that movement, ATPs are not being used because there is no way ATP will be there, right? It happens in nature all the time.
Diffusion.
Diffusion, yeah. osmosis. Okay. Now, passive processes include simple diffusion, facilitated diffusion, and osmosis. So those are passive processes. They don't require ATP. What is simple diffusion? Movement of molecules from high to low concentration. Movement of molecules from high concentration to low concentration, and that means towards the concentration gradient, towards the concentration gradient. Facilitated diffusion. You have seen that some integral proteins form channels, and those channels are always open. So the molecules, those are smaller than the size of the channel, will pass through that, right? You don't need energy for that. So that is facilitated. The protein is facilitating the transport. And osmosis. Osmosis is the movement of water. In bracket, you can write solvent. Water is the solvent. Movement of water or solvent from low to high concentration. From low to high. So, if this is a membrane, semi-permeable membrane, and you have highly concentrated glucose solution in this side. So this is glucose solution, okay? And in this side you also have glucose solution but low concentration. Okay? So this is low concentration, this is high concentration. When you make a glucose solution, how many things you have in that solution? 2. Glucose and water, right? Which one is solute? Which one is solvent? Water is solvent and glucose is the solute, right? Solute. So you have solute and water is the solvent. This is solute. Glucose. Same thing here. You have water and solute or glucose. Okay, now I told you that simple diffusion is the movement of the molecules from high to low. In this case, what will happen? Solutes will move from this side to this side because solute is more in this side, right? Is it clear? Solute is, glucose is more in this side, right? So glucose will move from this side to this side definitely. So that is what simple diffusion. Is it clear? Okay. Now, with this side, what is more? Which is more? Water is more. So water will move from this side to this side, and that is called what? Movement of water is called what? Osmosis. So when the water will move from low concentration to high concentration, that is osmosis, and when solute will move from high to low, that is simple diffusion. Make sense? Now just think, it will be very clear. What's the purpose of that movement? Why the things will move? To bring
the gradient.
Yeah, to bring the equilibrium, same concentration. That's the goal. So if you just think what's the goal, you can easily understand which one should move in which direction, right? If you want to bring same concentration, what should move from here to here? The solute, because glucose is more here, right? And what should move from here to here? The water. So one is simple diffusion, another is osmosis. Yes. I showed you that some proteins have channels, okay, and those channels always stay open, okay. So by forming channels, the protein is facilitating the movement. Make sense? Otherwise, water will not be able to pass through the phospholipid bilayer I have mentioned, right? So, who is helping? Who is helping the water movement? Protein, right? Protein is forming channel and helping the water to, otherwise water cannot pass through the cell membrane. So, facilitating, helping, okay? So, that's the example. This is just a test you can do in the lab. Take an U tube and separate by a membrane. In one side, if you take high concentration, in other side low concentration, that I have shown you here. Exactly same thing, right? So the solute will move from high to low, water will move from low to high. And finally, what will happen? In both sides, the concentration will become balanced or equal. Make sense? So you can do that test in the lab. In this one, you are testing only osmosis. How you can do that? You see now, if you put the semi-permeable membrane, which has very small holes, only water can pass, but not the solutes. In this case, what will happen? Only water will move from here to here, right? Because solutes cannot move. So the volume of this solution will increase, but finally the concentration will become same. Make sense? Volume has nothing to do with concentration. You see the concentration in both sides is same, although the volume is different. So that is another way you can test. Now the last thing, uh, tonicity. Uh, Tonicity is relative concentration. Tonicity is what? Relative concentration. That means what? You have a solution, and this solution has, for example, 10 milliosmol concentration. This is the unit of osmolarity. So milliosmol, 10 milliosmol is the concentration of this solution. Now, if I drop a cell, living cell, for example, red blood cell here in this solution, this is the red blood cell, and inside the red blood cell, the concentration is 5 milliosmol, for example. So this solution outside of the cell is hyper or hypo compared to this one. Hyper. Hyper means what? More, right? So the solution is hyper, hypertonic. Is it clear? Now, you tell me the concentration is 10 milliosmol outside and 5 milliosmol inside. Which way osmosis will occur? Water will move from always. Water always moves from which to which? Low to high, right? We have talked about that. So, what will happen? Water will move from low to high. That means from the cell, water will get out. Is it clear? So, what will happen to the cell? Get big or small? Shrink. So, that's the response of cell in hypertonic solution. Is it clear? The solution is hypertonic. Now, if I change this one, this solution to 1, now the solution is hyper or hypo? Hypo compared to this, right? So which way water will move? Always from low to high. So water will get in. Is it clear? So what will happen to the cell? Big, expand, swelling. So, or even may burst. So that's the response of the cell in hypotonic solution. Now if I change the solution to 5, is it hypo, hyper or iso? Iso means same, right? So no movement because balance is already there, right? So no movement of water will occur. So what will happen? Nothing. The cell will remain same. Make sense? So that is the response of the cell in isotonic solution. So if I ask you in the test that a living cell will respond, how a living cell will respond in those three conditions, now you can tell, right? Osmosis is the movement of water from low to high. Why we say tonicity is a relative concentration? Because you just think that I have shown you already. This is 5, and when it is 1, that means hypo, right? This is what? Hyper. Inside is hyper, right? Now if I change, keep this one same, but change this one to 10, inside is what? Hypo. So same concentration, but it is changing, right? So that's why you say relative concentration, tonicity. You are comparing two. If you change one, other might change. Okay, so we're done in cellular level. Good.